A new research reveals gene replacement therapy may heal a heart. Breaking a heart is difficult to deal with. Researchers in Japan believe that in certain circumstances, gene replacement therapy may be the answer. Research published in Stem Cell Reports in January found that heart cells from a patient with arrhythmogenic cardiomyopathy, a hereditary heart condition, do not contract appropriately when cultured in the laboratory, and that substituting the defective gene responsible for this impact corrects this problem. Welds between cells enable them communicate and move together in a coordinated manner, and abnormalities in genes involved in desmosomes cause arrhythmogenic cardiomyopathy. This gene, PKP2, produces a protein called PLAKOPHILIN2, which is essential for the formation of cardiac cells. According to the study's main author Hiroyuki Inoue, previous investigations in cardiomyocytes have demonstrated that mutations in PKP2 have a pathogenic role in arrhythmogenic cardiomyopathy. However, the cells employed in those studies were taken from healthy persons and were not tested for contractile activity. To find out how cells from patients behave in the lab, the researchers took a blood sample from a young person with arrhythmogenic cardiomyopathy, made some of the blood cells into stem cells, and then turned these stem cells into heart cells. They next created 3 distinct cell lines depending on how many mutant or intact copies of the PKP2 gene were present in the initial batch of cardiac cells. Cell contraction and desmosome construction were restored after replacing the mutant PKP2 with an undamaged copy of the gene, which the researchers could see using a time-lapse technique and fluorescently tagged desmosomes. Since our cardiomyocyte cell lines recreate the pathophysiology of arrhythmogenic cardiomyopathy, Higo believes that they may be used to produce gene therapies to treat the condition quickly and conveniently. Due to the prevalence of PKP2 mutations, new treatment techniques may be necessary to slow or stop the course of arrhythmogenic cardiomyopathy. To test novel treatments for arrhythmogenic cardiomyopathy, researchers may use the cell lines developed in this study. The study's results imply that gene replacement therapy might be an effective treatment option for people with this illness.